So I'm a medical oncologist. I work at the Christie in Manchester, which is a sort of standalone cancer hospital. So we and there's a couple of pictures there. The bottom right is our sort of treatment floor, and then we've got this new sort of research facility that finally has opened after a fire on the roof 7 years ago. And so this is the building on the left is where I'm sat now. And I also work with the research labs at the University of Manchester in our sort of Cancer Research Institute here in Manchester. And as I sort of said earlier, really, I'm a medical oncologist. I treat patients with head and neck cancers, but most of my practice is seeing patients with salivary cancers. But as a medical oncologist, I don't see patients who are kind of surgical candidates, i.e. people who present with a lump. You know, the first thing you do is you have a biopsy. That's normally done by a surgeon, and then they take it out. That's done by a surgeon. So that isn't really my patient group. I also don't see patients having radiotherapy because that's a radiation oncology discipline. So I actually only really see patients who have evidence of recurrent disease after their sort of primary treatment. I occasionally see people who don't, but that's more just for their kind of interest or involvement in our research rather than for any clinical need. And so just to start with some general kind of thoughts and descriptions around salivary gland cancer. So the purpose of today is to talk about acinic cell carcinoma. So acinic cell carcinoma is a type of salivary gland cancer. So what do we mean? Here's a picture I just sort of stole off the internet of salivary glands. You know, we have 3 pairs of salivary glands. We've got 6 we call them major salivary glands. The parotid, which is there just in front of the ear. The sublingual, which is, as the name suggests, under the tongue, and the submandibular, which is under the mandible, the jawbone. And you've got 1 on each side. So you've got 6 of these in total. And so these are the major salivary glands. Now, we also have what we call minor salivary glands, all these sort of small glandular structures all around the head and neck and even into the lungs. And then, in fact, we've got lots of other secretory glands, so glands that produce fluids, basically, like the tear duct or the tear glands, the lacrimal glands. And salivary gland type cancers actually arise in lots of different glands. But for the purpose of today, let's just keep it simple and say these are the salivary glands. Now, so to put acinic cell carcinoma in a bit of context, there are at least 24 different types of cancer that arise in the salivary gland when you classify them based on how they look down the microscope. It's what we call histopathology. So acinic cell carcinoma, as you can see, hopefully you can see the arrow. If you can't, then never mind. But in the box at the top here, this graphic has been generated from the Public Health England cancer registry data. So it is a little bit flawed cancer registry data. It's reliant on the information you put in, but at the same time, it's better than any single institution data set in that it captures, a larger number of patients from across the whole country. It's the English data set, not the Scottish and Welsh and Irish from our perspective. So it's only about 3/4 or 4/5 of our patients. And you can see, first of all, the top line, all salivary gland cancers. It's an old data set, the 4 years to 2013. But in that 4-year period, Public Health England only managed to capture 3,500, which I don't know whether to you that sounds like a lot, but it's really not a lot of cases. So the next thing to notice is this. So acinic cell carcinoma here, this big chunk of the major pie. So 16% of all salivary gland cancers were then subclassified as being acinic cell carcinoma. But there's a whole load of other types of salivary cancers. And so this is a challenge for lots of reasons. One, acinic cell carcinoma it's just showing you how rare it is. It's a small proportion of a small number of patients. The other is when we try and generate evidence for what to do. So to inform what kind of operation you should have, whether you should have radiotherapy, whether chemotherapy has any role, what other drug therapies might have a role. Most of the studies up until about 10 years ago would have just lumped together all of these different types of salivary gland cancer, or patients with all of these different types of salivary gland cancer, into one study. And then it turns out you can't really draw any meaningful conclusions. And so that's one of the reasons why we're still, I think, in the salivary gland cancer field, a number of years behind other more common cancers, probably a number of decades, behind cancers like breast cancer. So acinic cell carcinoma, though, as the take home, it's 16%, or about 1 in, whatever that is, 1 in 7 cases of salivary cancer, which in itself is quite rare. I put this in to try and reassure people. I think I put this in quite early on. So this is from that same data set. So from this Public Health England data set. So it's very top level data. It used this histopathology classification to look at what's described as relative survival, which means the proportion of patients who are alive. And pleasingly for patients with acinic cell carcinoma, their line is right up here at the top, which is a good thing. So just to help you interpret what this just describes is what proportion so if 100% of people are alive following, at the time of diagnosis, which you would expect, that purple line stays very close to the 100% mark. It drops down to about 90 to 95%, but that means most patients are longer-term survivors following a diagnosis of acinic cell carcinoma. And if you look at the other end of the spectrum, at this variant that is just described as adenocarcinoma, which possibly isn't really a variant, it's sort of it's just they just haven't been classified as having had any one particular type, have around only half of patients being alive at 10 years. So actually the prognosis, I would say, you could say is good. This, I'm not really sure what to make of this. It's useful when when you've got a rare cancer, you want to just look at what information is there. So this is again from that same cancer registry data set. It's the same purple line here, acinic cell carcinoma. And what's a bit odd, and I don't know what to make of it, is that if you look back from 1990 to 2013, the purple line weirdly goes up sort of decade by decade in terms of the frequency. So this is what they've labeled the age standardized rate per 100,000 population. And don't read too much into the number itself. It's a standard way that epidemiologists kind of state the frequency. But what's more relevant is just the trend. So a flat line, if you look adenoid cystic carcinoma, for instance, looks like the rate hasn't changed over 30 years or 20, 25 years. But the message actually is acinic cell carcinoma appears to be becoming more frequent. And if you look at it relative to its previous rate, it's quite significant. 0.04 to 0.16 is a sort of fourfold change. It's a bit odd. What's going on, I don't know. It may be just that it is becoming more frequent. There is an unknown risk factor that for whatever reason people are more exposed to. It may be that it's to do with diagnostic, skills. So it may be that it was underdiagnosed previously and is now being more accurately diagnosed. So maybe 20 years ago it was more likely to be just labeled as a adenocarcinoma, not otherwise specified. But actually this adenocarcinoma line, it does drop, but it doesn't drop that much. So it doesn't really fully explain it. So this slide, I've actually put in, I just quickly threw this in when everyone was introducing themselves, because I was aware that literally half of the room seems to be related to people under the age of 50. Yeah. So this isn't about acinic cells. So the purple line here isn't acinic cell carcinoma. And in fact, none of these lines are about acinic cell carcinoma. This is from the entire data set of 3,500 cases over however many years. But it just looks at people's survival by age. And what's interesting, this isn't a disease of young people. And the reason so in my experience, it's not a disease of young people, but I thought I'll just look and see what their classification. So here, all patients so 50 is the young cut point in this data set. And as you would expect, it's then grouped into decades going up to 80. And that's what we see, that the average age of patients would be around 70, 75, as with all cancers. So that was the reason I put this in. And in fact, so this is all salivary cancers, adenocarcinoma, everything else. Broadly, the younger people have a better longer term survival than all. Age in itself appears to be prognostic, but that's why I put this slide in. It's not really a disease of younger people. So this slide I've put in part just to talk about how people talk about these cancers, and there's a lot of terminology, so just to talk through, and then to think about how that might impact on survival. So we know that overall people do really well, but we describe stage. Okay, so stage 1, stage 2, stage 3, stage 4. So if you look here, just in simple terms, so this is that same figure as on the front slide, just about a salivary gland. So stage 1 is a small tumor in the salivary gland, nothing anywhere else, no lymph nodes. A stage 2 is a slightly larger tumor in the salivary glands, nothing anywhere else, no lymph nodes. And a stage 3 is a smaller tumor with lymph nodes in the head and neck region. And then stage 4 is anything else. And what you can see here is, so these are those sort of survival curves again, and it just shows people with early stage. So if you've got a small tumor confined to the salivary gland, you see your surgeon, the surgeon resects it, and for most people, you're cured. And this certainly pans out in my practice, because I very rarely see patients with acinic cell carcinoma. And as I said earlier, I only really see patients with recurrent or metastatic disease. So, early stage tumor, early stage acinic cell, very good long-term survival. But more advanced stage isn't. So, and this is my population, I suppose, those with metastatic disease. So staging is if you think about it, when you have the test done, you kind of have a biopsy and you have a scan. And broadly, staging is your scan information. It's like, how big is the tumor? Where is it? Has it spread to the lymph nodes? And that gives you your stage broadly. And grading is, you see these purple and black splodges. This is when you have a biopsy or a resection. The sample gets sent to a pathologist who looks at this and starts to describe it. And I've just put a few terms on here, but because grade is important as well, and broadly sort of low grade to high grade. And so your clinical teams will talk about these characteristics. So necrosis, I mean, they probably won't talk to you in detail about these characteristics because they'll assume you're sort of not really that interested, but it is useful to know what it is they're thinking. So necrosis is sort of areas of dead tumor in the biopsy. That's what necrosis means. Necro just means dead. So, and necrosis is something that you see in higher grade tumors. And you see that often because tumors, if they grow too quickly, they kind of outgrow their own blood supply, and they sort of, the tissue just dies in the middle. Fibrosis is a kind of scarring. They can lay down scar tissue, fibrotic tissue. Mitosis is cell division, so they look for cells that are actively dividing. They'll describe the borders of the tissue, so they'll look at, if they've resected a parotid gland, say, and there's a tumor within it, you can look at the kind of what they call the invasive front, is sometimes called. So it's like the edge of the tumor where the cells are sort of pushing, and you can see different types of borders, well encapsulated, well demarcated sort of borders versus invasive borders. This term about architecture, so sometimes they've got a very nicely sort of defined architecture, meaning structure, so you can see the different structures in there. And sometimes they're solid, meaning just cells lumped together. And then this term lymphovascular invasion, so the cells can go into, so invade the lymphatics or the vessels. And perineural invasion, so there are tiny microscopic nerves in the sample, and the nerves have a little sheath, and you can sometimes see cells around the nerve sheath. So when a pathologist looks at the sample, one thing they literally look at that's what these pictures are. I've just snipped them from articles, but this is what they look at, and they look for these sorts of things, and based on that, they'll give you a kind of grading. And then there was another paper actually very recently in the last couple of years in acinic cell carcinoma where they did the same thing, came up with their own kind of standardized, well, it wasn't standardized, in there they defined a way of grading, and they defined their cohort based on these things I've just described: mitotic index, necrosis, border, fibrosis, as high grade. And this mitotic index meaning number of actively dividing cells. So if it was more than 5 per 10 high power fields, and or there was necrosis, so dead tumor there, then either of those they described as being associated with higher risk behavior in their own right. So this was it was a big study, but a relatively small study. So it was about 130 patients. So in my mind, that's a really big study. But it is, you've got to caveat the conclusions in that, it's not quite enough to say with absolute certainty what it means, that this is the case. But in their 100 and odd patients, low grade, their 5-year survival was 100%. Now, 5-year survival is it's a surrogate for cure and long-term survival, but that's what they found in their cohort. So low grade do better. And so I think there is lots of evidence low grade acinic cell is a good thing. So grading is based on all this purple and blue stuff that their pathologists describe. I mean, this isn't my expertise. I look down the microscope, but this isn't part of my day-to-day practice to classify stuff, but the pathologist, that's their bread and butter, really, and they will classify something as high grade or low grade based on all these appearances. So I put this in for two reasons. One is you can see, so this is my patient cohort with salivary cancer. So I said I see patients with recurrent or metastatic salivary gland cancers. And so what you get here is, and this was 170 patients, 115, 140 patients, whatever that number adds up to when you add all those things up. So 115 in this thing, and then 16, 15, 8. So patients with acinic cell carcinoma, so over this period of time, which was a couple of years, we'd seen 8 patients with acinic cell carcinoma compared to 115 with adenoid cystic carcinoma. And what that reflects is that, because if you recall, I won't flip back through all the slides. Well, in fact, that's easy. If you recall on this slide, broadly, it's slightly higher, the frequency of adenoid cystic carcinoma, but they're a similar frequency. 21% of all salivary cancers are adenoid cystic, and 16% are acinic cell carcinoma. But then if you look at our practice, it pans out that, so from the cancer registry data, the message is the prognosis is much better. And I think, and from our practice, that's exactly what we see. So that's the first reason I put it in. And the second reason is just to introduce this kind of idea about genomics. So from the perspective of understanding the disease, I mean, there's lots of different ways to understand the disease. And in fact, it starts to get a bit sciencey, doesn't it, in a minute? So as an introduction to how we can understand the disease, if you think about how a cancer cell, what a cell is, what cells do in general, and what cancer cells do in particular, a cell is like a bag with DNA in the middle, and the DNA is kind of the genetic code. The DNA produces, essentially, it encodes lots of different proteins through a sort of middleman, RNA. So you've got DNA and you've got proteins, and the proteins broadly are how a cell does whatever it needs to do. And so you can classify cancers by looking at the DNA, because the DNA has often gone wrong. In fact, it's always gone wrong. They've never got a normal genome. And you can also classify it based on the RNA, the sort of middleman, and you can classify it based on the protein, which is a bit like the end product of this DNA. So, and the next few slides talk a bit about, I don't mean it's about RNA actually, but a bit about DNA and a bit about proteins. And the reason, oh, go on, someone said something. No. From my perspective as a medical oncologist, so I guess a surgeon's not going to change what they do in large part. If you've got a tumor that can be resected, a surgeon takes it out and they cure most people that they do that on. And so not much is going to change a surgeon's practice. For a radiation oncologist, it's useful to know more about the behavior of the disease, because it's not entirely clear, who is considered high risk enough to have radiotherapy, and certainly chemoradiotherapy. And I suppose we've got so few patients, it's not entirely clear who should be followed up in what way, with scans at what frequency, etc. So I think it is useful in their setting. For me as a medical oncologist, if I do have a patient with recurrent disease or if it spreads somewhere else, and we're thinking about drug therapies, then actually understanding as much as possible about the biology of that particular cancer is really useful, because when you don't have hundreds or thousands of patients that have been through studies before to define the standard of care, for me it's the best way to try and rationally both match patients to existing drugs and work out what drugs should be developed in this space, in this setting, in this disease context. And so some stuff is known about acinic cell. It's a little bit behind other cancers. It's a bit behind adenoid cystic. It's probably 10 years behind adenoid cystic of the salivary, and it's probably 30 years behind breast cancer. So one thing is this. So these are, when you do whole genome sequencing, you can get these these plots, Circos plots, circular plots that capture the whole genome for X chromosome, Y chromosome, and then all the other chromosomes going around. And about 4 or 5 years ago, there was a paper where they looked at just 4 patients, but found a recurrent gene rearrangement in this NR4A3. So there is one recurrent defining genetic alteration in the tumor. And for people who aren't familiar with this kind of idea about cancer genomics, it's important to be clear that this isn't a hereditary genetic alteration. So that it's not something that is passed down like from parent to child. This is a genetic change that has gone wrong in the cell in the salivary gland, and it's mutated and for whatever reason hasn't fixed itself, because most mutations should fix themselves, and it hasn't caused the cell to die. And in fact, it's done the opposite. It's caused the cell to survive and proliferate for one route or another. And so there is this recurrent gene fusion affects the gene NR4A3. And for Jeff, probably familiar with it actually, but it's very similar to the whole story in adenoid cystic carcinoma, the other common cancer type in the salivary gland that we see, in terms of why this causes tumor sort of growth and what the gene does. The other sort of take home about these circus plots is that there's a whole load of other stuff going on in the genomes. So all of these black lines represent other genetic changes, and so there are far more genetic alterations in patients with acinic cell than other salivary cancers, such as adenoid cystic carcinoma. So it's another paper from the last couple of years. It's a much bigger number of patients than we've got, although it's still a pretty small number. Looking at patients with acinic cell carcinoma, so most or all of these patients would have had the same gene fusion, but this was then looking for other. So this whole kind of list here is a whole load of other genes that have been looked at for other genetic alterations. And the take-home message was that there's this other gene, CDKN2A, which so the gene encodes a protein, well, a couple of proteins, but one's called P16, and it's a protein that's involved in what we call cell cycle. So a cell cycle is this whole cycle that cells do when they undergo mitosis. They divide, or well, they grow and then they divide, and one cell becomes two. And this whole process of one cell becoming two, that's a kind of fundamental problem in cancer. That's what cancers do. And CDKN2A loss, which is what is being seen in a group in about 40 or 50% of patients, is the consequence is uncontrolled cell cycle progression. And that's the sort of next commonest genetic alteration. I mean, it's not really known to be druggable. There are some drugs that target the sort of target of P16. The target of P16 is a protein called CDK4 and CDK6, and CDK4/6 is normally suppressed by P16. So this stops working. CDK4/6 is overactive. There are drugs, very sort of commonly used drugs, that inhibit CDK4/6, for instance. And this is like probably the biggest data set in acinic cell carcinoma. It's, for me as a medical oncologist, it's the biggest lead into the biology of what's going wrong or what's gone wrong in this disease. And it's the best lead we've got at the moment in terms of trying to rationally prioritize people for drug therapies. But again, just to reiterate, that this is my perspective is only patients who do have recurrent or metastatic disease. It's not for patients who've had a resection and are hopefully cured. This stuff is standard, so this standard staining. And then the next level that is standard is protein staining, looking for different protein markers where you can use those to confirm it's cancer and basically help confirm the subtype. And there's only about 10 proteins that you would look at. Then this is standard across all cancers, because it's not just for this. Beyond that, you could do whole genome to look for this NR4A3, but there's no point, because a pathologist will confidently say it's a acinic cell based on these purpley blue splodges, and the whole genome adds a whole extra load of time and effort and resource that doesn't change what's going to be done. When you get to my end of the thing, it does start to change what we do, but for most people it doesn't. You have surgery, and then you're followed up. So yeah, the level of analysis that's done typically stops after this sort of staining and protein staining, and everything else most other things would fall under research purposes. The patients we've seen, which is only a handful with acinic cell, but they may have had their primary tumor removed 10 years ago, and then for whatever reason they're back now, and that's why they're seeing me. So then we can call in their what we call archival, meaning stored, their archival tumor sample, and they're preserved. They're dropped in formalin, and that preserves them. So you do get some problems if they've just been stored for ages. Your tests aren't quite as reliable, reproducible, robust, but you can still get lots of useful extra information, and that's kind of what we do. That's what I do with our patients. We're kind of forever hassling path labs up and down the country to get tumor samples in. I think what I would envisage as like a dream would be if any patient in the UK and I only say the UK because it's I can't control what happens outside the UK. The world, we could say, would be better, wouldn't it? But if any patient in the UK who had a salivary gland tumor removed, if they were from the outset, everything was prospectively, in real time captured. So what are all their standard diagnostic things? What surgery did they have? What radiotherapy, if at all, did they have? What are all their other kind of whatever medical history sort of details? Do all of this up front, and then prospectively do it. It would be amazing. And it's not that impossible to do, I think, through a single healthcare system like the NHS. I think the world, it'd be difficult, but let's aim for the world, and then if we get it just in the NHS in England or something, then we can at least achieve something pretty big. I mean, that would be ideal. This was just to talk about, so again, this is patients with recurrent disease. And so this is just one figure from a study. And I'll just kind of outline. This was patients with salivary gland cancer who had recurrent disease, who had tumor profiling done, who had one specific mutation found in a gene called HRAS. And in fact, in this, I don't think HRAS even came up in this list. No, did it? No, I don't think it did. But nevertheless, this then shows the different subtypes. So these are the different subtypes of cancer, salivary cancer. And actually, acinic cell, there was only one patient, this AC. So in the study, they've called it ACCC, acinic cell carcinoma. From your perspective, your interest being acinic cell carcinoma, this is basically one patient. That's all we're looking at here. And this, but there were, whatever this is, a dozen patients who all had different salivary cancers, different types, different pathologies, but they all had one particular mutation. And so it's one of, in sort of oncology, medical oncology, kind of, future of drug development, one thing you might think is, well, could you have what you call tumor agnostic? So if you've got a HRAS mutation, doesn't matter what your pathology is, it's that sort of question. I think what this study showed is, well, not with this drug and this gene, is the first thing, because they had all different pathologies, they all had a HRAS mutation, and this shows the response. So change in tumor, percentage change in tumor size, if the bar goes up, that's a tumor growth, basically, is your best response. If the bar goes down, it's tumor shrinkage. But the best tumor shrinkage was this one patient with a parotid acinic cell carcinoma. So, it's only one patient. I really wouldn't read too much into it. I think I would read more into it, the principle of trying to match patients to sort of new drug therapies by tumor profiling. And I would say, though, that the signal is from this one patient, and maybe this patient here with a myoepithelial carcinoma, but there was also another here and another here with myoepithelial. So 3 were treated, 1 of 3 had a good shrinkage as their best response. And 1 of 1 had a good response here. And then this is, it's another example. I don't think this is an example that's really going to impact in the near term, but just the principle. So again, this was a paper from, I think it's a German group in the last year or so. So I said that before we can look at the DNA, the RNA, which we've not looked at, and the protein. And I was also saying, I showed those purple and blue slides, which are how pathologists look. So this is also the kind of thing, the pictures that pathologists look at. So they look at these purpley blue slides, and when they stain for proteins, they're always brown. So pathologists looking at kind of purple, blue, and shades of brown. And so in this paper, they took a handful of patients with different types of salivary gland cancer, and they looked for one particular protein, this protein CD138, which is a protein that sits on the cell surface. If you've got a cell like this, the protein kind of just sits there on the cell surface. And the job of that protein, I think, is to just kind of, it forms the sort of matrix around the tumor cells, and it helps tumor cells kind of connect to each other and speak to each other. And it's a tumor cell specific marker that's involved in cell connection. The reason it's sort of relatively of interest for you guys, I think, though, is so it's in principle, it's a marker that's a tumor marker. It's also in principle something that could be druggable. You might have a protein that sort of migrates to this, that has a chemotype drug on it, for instance. And this bar chart here says the percentage of this CD138 positive tumor cells. So acinic cell is here, so it's not saying 100% are positive. The middle part is only around 10%, with a range up to 25%. But nevertheless, acinic cell came out as one of the salivary gland types that had a higher expression of this particular marker. I think this last slide I've got. Everyone's sort of heard about immunotherapy. Well, you may not have heard about immunotherapies, but medical oncologists, immunotherapies are very exciting in principle. So in the last 10 years, immunotherapies, particularly targeting this PD-L1, so that's an immunotherapy sort of target, have been tried and tested across pretty much every tumor type, including salivary cancers. And in different tumor types, to different degrees, the expression of PD-L1, so PD-L1 is a protein, the expression of it in the tumor is used to sort of select out patients that respond. And this isn't really known for acinic cells, but in general in cancer, in lots of cancers, it's considered that those that are PD-L1 negative don't tend to respond. Those that are positive, it sort of depends how positive, and it depends on the tumor type. Anyway, there is data from a acinic cell that this brown staining is the staining. You can see it all just looks little splodges of brown, doesn't it? But pathologists will report, they'll call cells as positive, and then they'll say what percentage of cells are positive. And when 33 cases, a relatively small number of patients of tumor samples were looked at, all of whom had acinic cell carcinoma, around a third of them were PD-L1 positive by the standard criteria. So these are the, CPS means combined positive score, and it's the standard criteria we use in other head and neck cancers, for instance. So there is a signal that that a proportion of acinic cell carcinomas are PD-L1 positive, so may respond to immunotherapies. We don't really have good evidence of whether they do. There was a study, Keynote 028, looking at pembrolizumab. So pembrolizumab is I think I can probably confidently say it's the most commonly used cancer drug across all cancer types. Certainly in terms of how much the company get paid for this drug, they're the biggest grossing drug. Whether it's the most commonly used, it may not be the case. But it's a standard immunotherapy across lots of tumor types. And so this is the plot, the sort of response plot in PD-L1 positive salivary gland cancer. I actually don't know if any of these patients had a acinic cell, and if they did, it would have only been one or two of them. But the principle was it was patients with PD-L1 positive salivary cancers. And you can see that there is only it's not a strong signal. 3 of about 20 patients had significant tumor shrinkage, as in measured change from baseline going down here. But we don't have a study. If you had 30 patients with acinic cell carcinoma, which with this PD-L1 scoring gave all 30, I mean, that's again, as a medical oncologist, that would be the dream to have that study. But such a study doesn't exist. So that's the current sort of signal and information, I think, for immunotherapies in acinic cell carcinoma.